order, it went I, cap, promoter, operator, ZYA. I just remember, yeah, cap is after I.
There's that little I though first.
Good job. Thanks.
So let's just go ahead and assign what everything does. So I does what? I codes for what?
The repressor.
She's been working with
Yeah, repressor. The promoters is where RNA polymerase binds.
25 hours a week, and I was like,
This is the operator, it facilitates some transcription. This is beta gal. So that's the enzyme that separates lactose into glucose and galactose.
Yeah, that's how that works.
This is permease. And this one, who cares about it? Okay. So, the repressor makes the protein product, which is the repressor. That binds to the operator. It just always is transcribed for whatever reason. I think it has to do with this I. Whenever I learned this, I didn't really learn about I, but I think that has to do with regulating transcription of the repressor, which we always assume is present. Okay? So, repressor is always present, and it binds to the operator and stops transcription. And it's always bound there until the presence of lactose. So that's whenever the whole thing gets turned on, is whenever lactose is present. So if we call this lactose, this little dot or something. So lactose comes in, binds to the repressor, and it removes, because it changes its conformation that it's not able to bind to the operator anymore. Okay? So, say whenever lactose is present, the repressor is no longer bound. Okay? And whenever the repressor is not bound, RNA polymerase is able to physically come in there and begin transcription. So it starts on the promoter. Let's just draw something like that. So RNA polymerase binds and transcription starts. Okay? So whenever transcription starts, we get the downstream genes transcribed and then translated. So this will be our mRNA for beta gal and permease. And then we get our protein products. Like this.
I went into the final with an 85%, and I calculated my grade if I got 100 on it.
Okay, so what does beta gal do? Do you guys remember?
It was like 84.
It promotes, yeah.
Yeah, so beta gal is the enzyme that separates lactose into the monosaccharides, which are glucose and galactose. So I'll just go lactose to glucose and galactose. And permease, it's a transmembrane protein that's going to uptake more lactose from the environment. So if there's lactose, if you're exposed to lactose, then I guess the chances are there's more lactose around, and that's your energy source, you're going to bring more in. So I guess this goes and brings in more lactose. So it can metabolize more. So at this point, your glucose levels are rising. So increasing glucose has an effect on transcription as well. So there's another enzyme in the cytosol called adenylate cyclase. So do you guys remember what this one does? Adenylate cyclase? Right. So it takes ATP and changes it into cyclic AMP.
So yeah, 5.
And cyclic AMP complexes with cat binding protein.
Half between your intervals. So like if your X is from 0 to 5, and you had 5 intervals, you can only do 1, right?
So cyclic AMP will be like this little dot. Complexes with cap binding protein, and whenever camp is present, it binds here, and then only whenever this complex forms between cyclic AMP and cap binding protein can it bind to the cap site. So whenever that binds to the cap site, it kind of bends the DNA in a way that makes RNA polymerase easier to transcribe the downstream genes. So it upregulates transcription, you get higher rates of transcription. But what glucose does, so glucose is your final product of the whole thing in a way, as you're separating lactose into glucose and lactose. And glucose comes and regulates adenylate cyclase. So as your product is increasing, your transcription levels decrease by regulating this enzyme. So as glucose increases, adenylate cyclase activity decreases. Therefore, cap binding protein can't bind the cap, and you get only basal levels of transcription. And only until you run out of all your lactose will it rebind to the operator and everything stops.
Oh, wow!